Good morning, everyone. Yes, so I am the mouse guy, but like Sean said, I'm going to do my best to try to communicate some of these complex ideas. And so my senior project focused on the topic of dementia, which is something that's both personally and academically important for me. My time at COA has been spent developing an interest in neuroscience. And the reason that I'm so interested in neuroscience is because we know so little about the brain. And we know even less about how things go wrong in the brain when they do. And so what I wanted to do is tackle this topic of dementia from a number of different avenues. The first of which was actually a 5-week ethnographic project that I did in Europe, where I traveled to dementia research centers and communities in which dementia is particularly present, to try to get a sense of what dementia means for the people who will actually have it. What is having dementia actually like? And I wanted to make sure I understood this important aspect of the disease, or more accurately, a group of diseases, as I'll talk about in a few minutes, before I moved into a 6-week laboratory project at the Jackson Laboratory, in which I worked in Dr. Gareth Howell's research group. He focuses on neurodegenerative diseases, and I did a project there on Alzheimer's disease. That third portion of my senior project is endless reflection that will continue for the rest of my life on how to interface those two modes of understanding, a humanistic ethnographic approach and a scientific laboratory one. And I feel like I've learned a lot in that. So my colleagues at the lab have been constantly asking me about what I did in Europe and all of the great ethnographic findings that I gleaned from that project. So I'm going to take the chance here today to share with you some of my laboratory results, which they all know, but I really am excited about. And I want to start with a quote from somebody I talked to in Europe, actually. And that's, dementia is today what cancer was 20 years ago, and what AIDS was 40 years ago. And in this quote, I'm not talking about the incidence of the disease, when the world first saw these diseases, but more about when they were stigmatized. So the person who said this works with dementia and tries to improve a positive outlook on dementia, tries to share with the world the knowledge that people with dementia can be integrated into the society. But this quote is also really important because cancer and AIDS are both multifaceted, complex sets of diseases, along with dementia. And at those times, neither cancer nor AIDS had much of a cure. As of right now, dementia doesn't really have much of a cure. And dementia is actually a group of diseases, and Alzheimer's disease is just one of those. It is the most common form of dementia, and every day it's becoming more and more prevalent. And we know a little bit about the genetics of Alzheimer's disease. We've isolated a gene called ApoE, which is a gene that's constantly linked with Alzheimer's disease. And we know a little bit about how that relationship functions. And certain research groups are actually working on drugs currently to target ApoE and try to relieve some Alzheimer's disease symptoms. But there's another gene called BIN1, which stands for Bridging Integrator 1. But you don't need to know the name. All you need to know is that it's the second greatest risk gene for Alzheimer's disease, after that ApoE gene that I just talked about. We know that BIN1 is involved in formation of memories, in neuronal circuits, and in the specific Alzheimer's disease phenotype or symptom called tau neurotoxicity. And so BIN1 is seen in, or a certain variant of BIN1 is seen in a lot of Alzheimer's disease patients. But we really don't know how it works in the disease. So to understand sort of how genes work, what scientists often do is find a model and then knock out that gene, which basically means remove it from a mouse and see what happens. See if there are any important things that we find. It turns out that if we completely remove BIN1 from mice, they die shortly after birth. So it's very hard to study what happens if this gene is removed. The answer is a scenario called haploinsufficiency, which dumbs down to removing half of a gene from an organism, removing one copy of the gene instead of two. And so the question that we wanted to pose was, what is the phenotype associated with BIN1 haploinsufficiency? Or, for those of you who don't speak science, in English, what happens when mice have less BIN1 than normal? And this is important, one, in understanding how BIN1 is tied to Alzheimer's disease, but also potentially in creating a model for Alzheimer's disease research. If the mouse with only half of its normal BIN1 models well the symptoms normally seen in Alzheimer's disease patients, that might be a good animal to use for further research down the road. So we found a couple of interesting things. These are images of mouse brains, and on the left is an image from a normal mouse. On the right, a haploinsufficient mouse, only half of its normal bin1. These are sections of the brain in a region that's important for memory, called the hippocampus. And all those little white dots you see, each of those are contact points between two neurons, called synapses. The blue dots are the neuron cell bodies themselves. And so I hope you can see this in this image, but on the left, there are a lot more little white dots than there are on the right. So we see decreased synaptic density, fewer synapses in the brain, in mice with only half of their normal bin1. This is exciting because it could potentially explain some of the memory loss symptoms associated with dementia, associated with Alzheimer's disease. And we expect that when we behaviorally test these mice later on, that they'll show deficits in memory. This is that same region of the brain, the hippocampus, looking at just the thickness of cell layers in normal mice and haploinsufficient mice. Again, on the left is a normal mouse, on the right is one missing half of bin1. And so on the left, in the normal mouse, you can see the cell layer here. Each of those dots, again, is a cell. It's about 5 to 6 cells thick on average. On the right, only 2 to 3 cells thick. So we see a pretty drastic reduction in thickness in this area of the brain that's important for memory formation. And that's an exciting result because it actually correlates really closely with what we see in the clinic, in Alzheimer's disease patients, in humans. We see drastic reductions in hippocampal volume. That size of that region just becomes smaller. So this is an exciting finding that might indicate that these mice, missing half of their bin1, will be good models for Alzheimer's disease. We don't, however, see the classic symptoms of Alzheimer's disease, which are related to inflammation and immune response. On the left, and stained in red, are a type of cell called astrocytes that are involved in neuronal maintenance. And an increase in the number of astrocytes can be indicative of an inflammatory response. On the top is a normal mouse, on the bottom, that haploinsufficient mouse. And you can see there's not really much of a difference. On the right, in green, are a type of cell called microglia, which is the brain's immune system. And so if there were to be more microglia, we would expect that those mice are having a greater immune response, which is something that is seen in Alzheimer's disease patients. Again, no change between these two types of mice. So the bin1 haploinsufficient mouse doesn't model Alzheimer's disease perfectly, but we do have some exciting results. Again, that lower synaptic density in the hippocampus, that region of the brain important for memory, that could possibly explain the memory deficits associated with Alzheimer's disease. And the thinning of hippocampal cell layers, which align with what we see in human Alzheimer's disease patients. The fact that there is no inflammatory immune response could, again, mean that maybe this mouse doesn't model Alzheimer's disease that well. It could also mean that bin1 isn't involved in the breakdown of the blood-brain barrier, which is the separation between the blood and the other cells in the brain, which is seen to degrade in cases of Alzheimer's disease. It could be that there's another gene, potentially ApoE, potentially something else, that's involved in that component of the disease. So the sun is unfortunately setting on my presentation, like it was in this beautiful day in Amsterdam. But I want to take a little bit of time right now to tell you where I'm going to go after I leave COA. I'm actually going to spend the summer working in Dr. Gareth Howell's group at the Jackson Laboratory, and I want to thank him right now for all of his help and for showing me the ropes on how to study Alzheimer's disease in the lab. And then I hope next year to apply for a research assistant position in his laboratory. I'd also like to thank my other project directors, Drs. Darren Collins and Chris Peterson, for their incredible help and support throughout my entire project. And thank you for putting up with my scientific jargon vocabulary. I really appreciate it. Thanks.